Hello everyone, thank you for coming. Before we start, I would like you to complete a survey. This will be the URL right here, and it's basically just a question, if you could pick the traits of your future child, would you? And it'll be like, yes, no, I don't know enough information. On August 6, 1945, the first atomic bomb was dropped in Hiroshima, Japan, causing thousands of deaths and devastating destruction. Once the knowledge on how to create such a weapon was released, it could never be taken back, and now the reality is 9 nations today continue to harbor nuclear weapons as part of their self-defense network. This acquisition of knowledge happened to release a Pandora's box filled with massacre and endless paranoia. Recently, scientists have found a tool that could change humanity's future, but for the worse or for the better. One thing's for sure, once this technology is used to edit our genes, the world will never be the same. Let's just hope the result of nuclear weapons doesn't foreshadow the possibility of genetic engineering in human beings. Hello, my name is Elaine Thutt, and I am here to talk about how not all progression should be embraced wholeheartedly without thinking about the consequences. Genetics are the puzzle pieces that make up each and every one of us in this room. Who are we to rearrange the final product? To reach a form of compromise in which some benefits of gene editing can be enjoyed while most of the risks are avoided, only certain areas of genetic engineering in human beings should be explored and tested until they can safely be applied using modern-day science, while other areas should be banned entirely to ensure humanity doesn't become too wrapped up in controlling their own evolution. To give you a little preview about what we're going to talk about, first I'm going to give a small science lesson on what genetic engineering is and the different subcategories. Next, I'm going to go over the recent advances, where the technology is now. After that, I'm going to go over kind of the hype. Why is this so important? Why will this change our future? And why do we want it to work? And after that, I'm going to talk about what's stopping us, and that is essentially morality. And after that, I'm going to talk about alternatives. Okay, so before we delve into a moral debate, let me explain exactly what genetic engineering is, the different categories you need to know, and why. Genetic engineering can be defined as the editing of an organism's genes in order to manipulate a certain trait. We see the results of genetic engineering every time we walk into the produce section of our local grocery store. I know you all see the tomatoes over on the rack that are vibrantly red and huge. We can automatically assume that the tomatoes did not naturally grow that way. A scientist, most likely, edited the genes of the tomato plant to make to amplify some traits to make the product as appealing to consumers as possible. The two categories within genetic engineering are somatic cell gene therapy and germline gene therapy. So first we're going to talk about somatic. These are just essentially different types of cells that DNA can be taken out of to edit. So a somatic cell is any cell in your body that is not a reproductive cell, which are eggs and sperm. Editing the genes of a somatic cell affects only the individual the changes are made in. Germline cell therapy has to do with germline cells. Germline cells are reproductive cells, so eggs and sperm. And once you edit the DNA in a germline cell, the changes are passed down to all of your descendants, so your entire progeny. When learning about recent issues in genetics, specifically germline gene therapy, so the one where your DNA is edited and it's passed down to all your descendants, a phrase you will commonly encounter is genetic enhancement. It should be noted that genetic engineering and genetic enhancement are two different things. Genetic enhancement is taking the methods of genetic engineering and using them for things other than medical cures. It is the idea of improving traits such as height, strength, intelligence, etc. Oftentimes, genetic engineering will be referred to as a slippery slope because people believe it will start off with good intentions, like the idea of curing like genetic diseases, but will eventually become a tool we use excessively for the unnecessary. For example, editing your baby's traits before they're even born. To me, editing human genes sounds like some fictional dystopian future that is thousands of years ahead of us. So why are we talking about it now? We are talking about this now because a new technology was discovered called CRISPR-Cas9, which enables scientists to cut and paste sections of DNA more efficiently and more accurately than ever before. This means that the future I thought was thousands of miles away could actually be right around the corner. Here's a video to explain what CRISPR does from Jennifer Doudna, the founder of CRISPR itself.
A few years ago, with my colleague Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to defuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells.
All right, we're going to stop there. But one thing I really want to highlight, which she said at the very end of the video in case you missed it, is that human embryos have already been tested on in China. So on April 18th of last year, a study was published from a group of Chinese scientists stating that the findings after testing on human embryos with CRISPR-Cas9. Although the embryos were non-viable, meaning they couldn't eventually become babies, a debate was still sparked by the public. Some thought an ethical boundary had already been crossed, but we will get into that much later. What they really found is that CRISPR isn't ready for the advanced measures they would like to take. Which leads me to my next point. But wait, CRISPR is not, sorry, I didn't mean to alarm or excite you, and I'm sorry if I did, but the truth is CRISPR really isn't ready to use in humans right at this moment. When tested in complex organisms, it tends to have unpredictable results. In this specific experiment, the scientists attempted to use CRISPR to remove a gene that causes a fatal blood disorder. It was performed on 86 embryos. Out of that, only 28 had the gene cut and removed correctly, and even fewer incorporated the new healthy gene. So we still have a long way to go testing and making sure that it's safe. Eric Lander, a geneticist, another point as to why we're really not ready to use CRISPR is that it's only been a decade since the human genome was even read. Eric Lander, a geneticist and professor of biology at Massachusetts Institute of Technology, wisely states in his article called The Brave New Genome that it has only been about a decade since we first read the human genome. We should exercise great caution before we begin to rewrite it. The scientific field focused on DNA and genetic engineering is incredibly fast moving. Look how far we've come in genetics research just within a decade. But this also alludes to the fact that we still have much to learn about DNA itself. It is hard to correctly edit and manipulate something you don't fully understand. And the last part here, hashtag CRISPRFacts, is actually a hashtag on Twitter was created to try and tone down some of the hype that was made over CRISPR because it was such a big deal that people were getting so upset about it. So people made some jokes about CRISPR and would use the hashtag CRISPRFacts to kind of tone down some of the hype. But one thing to note is that CRISPR may not be ready for correcting human deficiencies yet, but it could be in the near future. So we should talk about ethical boundaries now, so society is prepared for when it does happen. So, why do we want this technology so bad? What are the benefits? The reason that it's so hyped up is that it could really be a cure-all. The main reason scientists are even examining gene therapy is the hope that it can cure genetic diseases and help us gain better understanding of more common diseases. Genetic diseases like sickle cell anemia occur because of a defect in one gene. If CRISPR-Cas9 could be used to alter this gene, we could have a cure for disorders like this one in Huntington's disease and cystic fibrosis. Editing germline cells of people afflicted could remove the disease from humanity's entire gene pool, so we never have to deal with it again. Another exciting hope for the use of CRISPR in humans is that this will help us make connections between certain genes and diseases such as Alzheimer's and cancer. Before CRISPR, this process was a slow form of trial and error of testing in animals. Now the process can be sped up and the connections found much faster. However, these harsh diseases may contain roots in hundreds of different genes, making it more difficult to foresee whether gene editing could offer an eventual cure. And lastly, this one is a benefit not really related to current medical problems, but it's still cool nonetheless. It's this idea that CRISPR could create a human race that could think quicker, run faster, climb higher, and feel deeper, and could lead humanity to stretch beyond its physical and cultural limitations that are biologically in place now to become more improved in almost every sense of the word. By using genetic enhancement, we are reaching humanity's full potential and alleviating unnecessary human suffering, according to Nick Bostrom, a professor in philosophy and a director of the Future of Humanity Institute at Oxford University. So why aren't we utilizing this now? Why isn't everybody on the bandwagon, so to speak? Some people are concerned. The main objections to this have to do with morality. So what are people concerned about? The first would be to take this step of engineering our own genes is to take human evolution into our own hands. It is essentially playing the role of God. Our species would be making monumental decisions on which genes in future generations survive and which die out. Genetics would never be up to chance again. This can go one of two ways. It can make a race of superhumans or create a future dystopia where genetics is used as a weapon in the form of super soldiers. So, who knows, it's kind of that risk versus reward idea. Another moral objection is the idea of the loss of the individual. And that would be like, if selecting children's genes ever becomes a reality in the future, not only genes for physical traits would be determined, but also personality traits. And a large part of the human experience is finding and defining oneself as an individual. If parents picked out its child's personality traits, this journey of self-discovery no longer holds the same meaning. So are we losing kind of the sense of who we are if somebody is engineering us to be a certain way? Another objection is that there is seen to be a loss of freedom. Concerning the child specifically, the children subject to genetic engineering may feel as though they have lost the freedom to create their own identity as an individual. The child's freedom of choice would be detrimented. Genetic enhancement decisions made by parents would end up steering their children towards certain life choices. So that kind of goes hand in hand with the loss of the individual. And the last would be a loss of equality. Genetic engineering would introduce a whole new way to discriminate, and that would be the genetically engineered against the natural human beings. And since the idea of genetic enhancement, engineering your own baby's traits, would probably be a fairly expensive procedure, you'd kind of have that line drawn between rich and poor. So you're just redefining, or making the line deeper between the rich and the poor, and to discriminate against each other. But not all of moral arguments are objections. There is one moral argument specifically from an Oxford professor of philosophy, named Julian Savulescu, who takes sort of the opposite approach. Julian believes that if we had the technology and knew that it was safe, it is our moral obligation to make our children the best they can be. So it's kind of a devil advocate's point of view. Now lastly, we're going to talk about alternatives. Is genetic engineering really necessary if we have different ways that, this is mostly concerning people who are concerned with having a healthy child if they contain recessive genes for genetic diseases? Is genetic engineering really necessary for these types of people if there are alternatives for having children? There's this thing called PGD screening, which you fertilize maybe your egg and your partner's or your spouse's sperm outside of your the body, and you screen each embryo for the one that is least susceptible to the specific genetic disease you are looking for. So that's a way to try and avoid certain genetic diseases. Another idea is third-party reproduction. I'm sure you've heard of it. It's has to do with a sperm donor or an egg donor. If you're getting your genes from somewhere else, combining them with your own, then if you're picking certain genes, you're less likely to get harmful ones. And lastly, adoption, which is not biological, but is still a viable option. Lastly, I'm going to talk about my service learning. This is basically how I've taken all my research this year and applied my knowledge to something that I believe is useful. This year I wrote an editorial to advocate for my opinion to ban germline engineering. And that's kind of what I was talking about earlier about advocating, or taking the side of one part of genetic engineering but banning another. I want to ban germline engineering. And I have submitted it to the school newspaper, The Highlander, which will be released at the beginning of May. I am also currently attempting to submit it to our local Birmingham newspaper, so if you are all subscribed to the Eccentric, keep an eye out. Remember, editing germline cells means the changes are passed down to every descendant for forever. This means no mistakes can be made. Edits stick. Although the rewards of gene editing are tempting, the risk that comes about because of germline editing puts a damper on the excitement. This doesn't mean I think the field should not be explored and tested in more. I fully support the testing and editing in somatic cells. This way, some of the benefits can still be achieved while risking a whole lot less. In conclusion, we'll just do a quick summary of what we talked about. We talked about somatic cell engineering and versus germline cell engineering. In somatic cell engineering, though it may not be able to offer a permanent solution to certain things, I think that it is the much safer option to explore versus germline engineering, where edits are for forever. And next, we talked about the improvements that that genetic engineering could make in the human race, improving it with genetic enhancement, or just looking for cures for genetic diseases, making connections for common diseases. After that, we talked about morality, what's stopping us from using it right now, moral objections, basically. We believe that it's difficult to have someone decide which genes are good and which genes are bad, and everyone will have varying beliefs on that. After that, we talked about different alternatives that you could do instead of using genetic engineering, which are all viable options and all should be considered. So thank you very much for listening to my presentation. If you have questions, I will take them now.